Sanchez Ramos.
Thank you. Good afternoon. Normally there's questions, but I just wanted to get some information to you hot off the press. It's always quite interesting. This I found in my box yesterday. It's a movement disorders journal. It's published every 2 weeks. And it has the latest findings either in basic research related to Parkinson's disease or clinical research. This one is very exciting to me because many people ask me, will we ever have a cure for this illness? And I said, well, you know, there's so many kinds of Parkinson's syndromes. For some of them, we know how to cure it. If it's a Parkinson's syndrome that's due to a medication, we just stop the medication and people get better. But that doesn't happen too much nowadays. Only psychiatrists tend to produce Parkinson's syndrome because some of the major tranquilizers they use block dopamine receptors and cause Parkinsonism. But they're more and more educated about it, and it's a dose-dependent thing. And so the classic tranquilizer that caused Parkinson's syndrome was called thorazine, or phenothiazine, or Haldol. These were drugs used to treat psychosis, and they still are used. They're used to treat aggressive, violent behaviors. They're called major tranquilizers, to be distinguished from the minor tranquilizers that are the #1 drugs of abuse, illicit drug abuse in the United States among people 60 and older, the minor tranquilizers. They don't cause Parkinsonism, but they do cause depression, sedation, and dependence. That's Valium, Librium, etc. So there are major tranquilizers and minor tranquilizers. Minor tranquilizers are the preferred drug to be prescribed by family doctors for anxious people or people who don't sleep, because a lot of the minor tranquilizers are to last a little bit longer, and you take it at night to help you sleep. Anyone who sees me knows I hardly prescribe those drugs. There are better things to use than that. And they tend to exacerbate depression. But I'm going to get back to the Parkinson-inducing tranquilizers. And the only reason I'm bringing it up is for twofold. One is it's the one situation where we can make the Parkinson syndrome disappear. Just take away that drug, thorazine or Haldol. But the other bit of information that was important about it is when this was noticed in 1950s, when these major tranquilizers were first introduced into practice for psychosis, research on how the tranquilizers worked led to insights about dopamine. So they began to realize that there was a dopamine problem in Parkinson's disease, and that led to the development of levodopa, or Sinemet, which is a way to replace dopamine. And we focused since 1960 on dopamine as the major issue. And we know there are environmental poisons and toxicants that can destroy dopamine-producing neurons. Those are not reversible, unfortunately. Luckily, there aren't that many people who suffer toxicant-induced Parkinsonism. However, exposure to these environmental toxicants, some types of pesticides or herbicides, are known to increase the risk for Parkinson's disease, just as certain heavy metal exposures. So people who work in battery factories or who work as welders are exposed to a lot of heavy metals, especially manganese, and that increases the risk for Parkinson's disease. So you can maybe not cure that form of Parkinson's disease, but you might be able to avoid it by making people aware that these occupations have a higher risk of developing Parkinson's disease. So I'm getting at what's really exciting here. So there are some kinds of Parkinsonism we can avoid, some we can reverse, but most Parkinson's disease, well let's call it Lewy body Parkinson's disease, and I'll explain what I mean by that, is of unknown cause, and we don't know how to cure it. But since 1930, we've been aware that the brains of people who when alive, who had Parkinson's disease, when their brains are examined after death, systematically, they find these funny kinds of inclusions in cells. So you have to look at the brain under a microscope. You do certain kind of staining procedures to visualize these spherical inclusions the guy who described them was named Lewy, so they're called Lewy bodies. They're intracellular accumulations of proteins and lipoproteins. It's fatty material conjugated with proteins. And the proteins are impossible to digest. Most proteins can be broken down, just like a steak. You put it in your mouth, your acid breaks it down, then enzymes break it down into its constituent amino acids. And each cell has a system for breaking down proteins to be recycled into its component amino acids. I'm giving you all this biology lessons, but it's so you'll understand what a Lewy body is. Lewy bodies are considered the hallmarks of Parkinson's disease. In other words, if you really want to make sure that you had Parkinson's disease, the really convincing way is after you die. There are other ways that we do that are very good, that are 90% accurate, 95% accurate. But the final diagnosis comes too late. That's what we say, that the internist knows everything and cannot really do that much. The surgeon knows nothing and thinks he can do everything. And the pathologist knows everything too late. So the Lewy bodies are made of these proteins that can't be digested. We call them misfolded proteins. And those misfolded proteins are very characteristic of a whole family of neurodegenerative disorders, where there are populations of brain cells that start waving goodbye. They start not functioning and then they disappear. They undergo a gradual cell death. In Parkinson's disease, most of those are dopamine neurons that are waving goodbye. And that's where you see a lot of Lewy bodies, in those dopamine neurons. In Alzheimer's disease, there's a region of the brain called the basal nucleus of Maynard, and those neurons start accumulating proteins, abnormally folded proteins, called tau or neurofilament proteins, and they're impossible to break down, and they accumulate. They also, in Alzheimer's, also accumulate something called amyloid that's outside of cells, and they it's a little bit different than the Parkinson's disease. In Huntington's disease, there's an abnormal protein that accumulates called the Huntington protein. And then there are these really weird disorders where all it is an abnormal form protein that is transmissible slowly. And that is something you've all heard of. It's Creutzfeldt-Jakob is the human condition, but it's very much like mad cow disease. And it comes from prion diseases. And that's the prion diseases are very strange, because all a prion is, we all have prions, which are just proteins on the cell membranes. But if the protein assumes an abnormal shape, it forms an abnormal folding. Because proteins are just a sequence of amino acids like rosary beads or prayer beads or a necklace. But you know how a necklace, when you put it down, it forms a three-dimensional. You take a clump of beads that's strung on a chain, because that's what all a protein is a string of amino acids. But depending on what the amino acids are, it'll take a three-dimensional structure that's unique. Imagine you have a pearl, then you have like a jagged rock, and then you have a tiny little bead. And so the sequence of the amino acids is made up of these 23 amino acids in various proportions. But if some of them have some weird sticky stuff on them, when you put the beads, that chain of beads in your hand, for example, it'll form a three-dimensional looking thing. Say the 10th bead will stick to the 20th bead normally, but if there's an abnormality there because of mutation in the sequence of the protein, you'll have an abnormally formed, a misfolded protein. Every protein has a sequence of amino acids that will determine its three-dimensional shape, and that three-dimensional shape will determine the function, the enzymatic function. So when I talk about proteins, and these are things that you learn when you go to medical school or college in biology, and many of you never did that. But the reason I'm telling you this, because the answer to all of these neurodegenerative diseases, the big clue is that all of them consist of an accumulation of abnormally formed protein. Why do they abnormally form this three-dimensional structure? Well, we know, and get back to the Parkinson's disease, that there are mutations in a protein called alpha-synuclein that result in an abnormal accumulation of alpha-synuclein in the Lewy bodies. So a major component of that Lewy body that was discovered in 1913 is called an alpha-synuclein misfolded protein. Now, alpha-synuclein in its normal shape is a very important protein found on the synapses of neurons, very important in the communication of one neuron with another, because that's what our brains do, is a network of communications pathways. So, simplify again. So in Parkinson's disease, in all cases of idiopathic Parkinson's disease, in which we don't really know the cause, there's an accumulation in the brain of Lewy bodies. Now, we used to think it was only in the dopamine neurons. But in the last, I guess 10 years, we've learned that those Lewy bodies originally appear in the olfactory bulb, which is in the very forefront of the brain. That's why people tend to lose the sense of smell early on, before they have Parkinson's disease. And in the brainstem, way below the midbrain. And in the brainstem, in the part of the brainstem that receives the vagus nerve, and the vagus nerve controls the gut. So that's where the Lewy bodies first appear, and they may be associated with some constipation and loss of sense of smell. Those would be prodromal to Parkinson's disease. Those Lewy bodies gradually disseminate, they spread. And how they spread is a hot area of research. When they get to the midbrain, that's when the first symptom of Parkinson's disease appears. So the process starts for 10, 15 years before you actually have the first little bit of tremor of your little finger. So that by the time you make the diagnosis, people have lost quite a lot of dopamine neurons. And in those cases where those Lewy bodies spread, not just from the front to the midbrain or from the bottom to the midbrain, but to the cerebral cortices, the highest level of brain evolution. That's what gives us our ability to think, to write poems, to plan ahead, to abstract, to do mental arithmetic. Those higher cortical processes become involved in a form of Parkinsonism called dementia with Lewy bodies. It used to be thought, before we knew much about Lewy bodies, that dementia that was out of proportion to the Parkinson syndrome was Alzheimer's plus Parkinson's disease, which, by the way, can happen. But dementia with Lewy bodies is recognized more and more as one of the dementing illnesses, and with the Parkinsonism being the least component of it, so to speak, because the dementia is much worse. And I've heard that Robin Williams supposedly had dementia with Lewy bodies, and one of the characteristics is that they start hallucinating, seeing things that aren't there way before they take any medicine. Hallucinating when you take levodopa or other medications for Parkinson's is well known to happen, usually, let's say in about 20 to 25% of patients, usually who are having problems with sleep and or are having cognitive decline. And then those are the ones that tend to have hallucinations. But dementia with Lewy bodies, which is attributable to involvement of the cerebral cortices, the frontal, parietal, and temporal lobes, you have three regions of cerebral cortex. When those are involved, that's dementia with Lewy bodies. So we're getting to the big breakthrough. I'm just giving you, because if I told you what the breakthrough is, you wouldn't understand how this could be a cure. What I'm trying to get you to understand is how important the Lewy body is and its spread. Because it's its spread of the Lewy bodies in normal Parkinson's disease is what accounts for the gradual worsening of the illness. You know that people with Parkinson's disease don't stay the same. The symptoms gradually get worse. And it's associated with spreading of the Lewy bodies, and then as they get more and more neuronal systems, you get more and more symptoms. So the fact is that if you take Lewy bodies from a human, it has a mutated alpha-synuclein, or abnormally formed, because the mutation of alpha-synuclein doesn't really occur in that many people with Parkinson's disease. There's something happening that causes what looks like a mutation. In other words, it's not running in the family like the original alpha-synuclein mutation, which is a dominant disorder passed on to every generation. Most people with Parkinson's disease don't have that, but they have an abnormal alpha-synuclein. And could it be a toxic agent that causes the abnormal formation? No one knows, but we do know that if you were to take abnormally formed alpha-synuclein and inject it into just the cerebral cortex of a mouse, it's a healthy mouse, over time, that alpha-synuclein forms the Lewy bodies and it spreads throughout the mouse brain and it starts causing Parkinson's disease. This is almost like an infectious process, like prion disease, but it's not a virus or a bacteria. Just having an abnormal formed alpha-synuclein will cause these accumulation of these materials called Lewy bodies, and they gradually spread over in a mouse pretty quickly, in humans over decades. So if we could prevent the accumulation of Lewy bodies and their spread, that'd be a cure. So this is what's really cool. I'll read you the title of the paper. siRNA delivery reduced alpha-synuclein aggregates in a mouse model. It's in a mouse, but it's very exciting. Now, I'm going to have to tell you another big development. In other words, there's a new drug type called small interfering RNA. I might have talked to you about it. Certainly, I've been working with small interfering RNA as a way to stop gene expression in Huntington's, where it's one mutation causes an abnormally formed protein called Huntington protein. And if you can prevent that DNA from expressing the protein using what's called small interfering RNA, you can prevent Huntington's disease. And we can do that in mice. It's just the problem is delivering it to brain throughout life. You got to continuously bathe the brain with this drug, with this molecule called small interfering RNA. The small interfering RNAs are bigger than most drugs in terms of weight, molecular weight, and they can't be given by mouth or systemically. They're digested. So the big challenge is how to deliver small interfering RNA, which will prevent certain genetic disorders. So when I read this, I said, well, it doesn't make any sense to me here. Why? How could Parkinson's disease that has accumulation of alpha-synuclein, how could you prevent the spread of these Lewy bodies with small interfering RNA that's directed against the expression of alpha-synuclein? And it turns out that's what they did. There's a lot of technical work on it that I'm not going to describe, but there are two important steps that they technically solved. One is to prepare a nanoparticle. A nanoparticle is 100 to 200 nanometers. A nano is a billionth of a meter. Think of $1 billion. Just take one, that's one. A billion, 10 to the 9 zeros. It's too big to really contemplate. 10 to the 9th and 100 10 to the 9th is the size of these particles. And they make these particles that they call exosomes in a cell culture and they load them with siRNA that's a very particular kind that will recognize the expression, the messenger RNA that makes the mutated alpha-synuclein. So even though it's not a genetic disorder, there's still this process of DNA being made into messenger RNA being made into the protein. If there's an abnormal sequence from the abnormal protein, you can prevent the accumulation of alpha-synuclein. So this paper describes the successful preparation of a drug that they give intravenously that is really nanoparticles they call exosomes that contain a new kind of drug called siRNA against alpha-synuclein. And so that seems to prevent the spread of the alpha-synuclein in the mice, and it reverses symptoms that had already begun. So it's very clear that the accumulation of alpha-synuclein is responsible for the symptoms and signs of Parkinson's disease. And we already kind of knew that, and we didn't know how to prevent alpha-synuclein from spreading. So this complements a second approach that could cure this disease. This is very exciting for me because in my lab, we're developing nanoparticles to deliver small interfering RNA against gene expression. but looking at a model of Huntington's because it's one gene. But now, maybe we're going to start looking at using our delivery system. Our delivery system is actually easier because you don't have to inject it. You don't have to. The way they prepare this in exosomes is really complicated, but the way we deliver it is through a nasal spray. So I think we'll do better. So it'll be easier to take a nasal spray once a week and get that small interfering RNA against alpha-synuclein into the brain and thereby prevent the spread of the alpha-synuclein and slow progression, maybe reverse some symptoms if you start early enough especially. So that's very, very exciting. Now the other way that we can cure or prevent Parkinson's disease is to develop a vaccine against alpha-synuclein. And that Parkinson Research Foundation actually gave a small grant when I first started doing this, to a colleague who was developing a vaccine against the alpha-synuclein. And unfortunately, we never got larger amount of support. People are still working on this, and we may get back into this business as soon as we have several $1 million to devote to this research. You never know, I could win the lottery. It's actually a matter of having the right kind of reviewers review your grant. And, in order to get an NIH-funded grant, it has to be in the 7th percentile. In other words, there's a 7% chance of getting the grant. There's a 93% chance of not getting it, so you got to keep on plugging. And oftentimes, the people that get those 7%, they get those grants, are the same people that have been getting them all along. So there's a lot of difficulty now. A lot of people believe that it's better to get a major angel investor to invest in a very exciting project. Once you have a lot of data, then, in other words, you do the research. This is how NIH works. If you've already shown that it's working, then they'll fund it. It's but they won't fund anything that they think is too risky, that you're not going to get any data. So getting pilot data is very important. And so that's what we're working on with our new delivery system. What was really cool is that a lot of people are thinking the same way. We've got to harness small interfering RNA. I'll tell you a bit more about what small interfering RNA is all about. The cell, every cell has a nucleus, and that's where all the master molecules are stored, DNA. And DNA is constantly being decoded into a messenger RNA. You don't want to mess around with the storage, the archive of information that determines who you are, that determines you. So you make a copy of a gene, and that's called messenger RNA. That messenger RNA is kicked out of the nucleus into the cytoplasm. It's within the same cell, but outside the nucleus. And there, it's translated into the protein. And then the protein can be a structural protein that make like keratin or a neurofilament, or it can be an enzymatic protein like tyrosine hydroxylase. So that's the essence of molecular biology that we learned in colleges. DNA has master code, makes messenger RNA, which it's going to be degraded after it's used, and then the messenger RNA is made into a protein. The proteins are exported to where they're needed, and they're broken down and recycled so you can use those building blocks again. But the original DNA is not changed. If you have a problem with the original DNA, because you got radiation, you can get mutations and abnormal proteins and demise. So that's the key. Now, so we knew there was messenger RNA, DNA. There's also something called transfer RNA. There's the newest kind of RNA called small interfering RNA. And those were discovered in the 90s. They were first discovered in plants. And it was a botanist who found that certain viruses that infected plants, they were called RNA viruses because their nucleic acid was not DNA, but RNA. In fact, AIDS virus is an RNA virus, but many plant viruses are RNA viruses. The DNA viruses are like smallpox virus. Ebola virus is a DNA virus. In other words, the messenger, the DNA, the code that determines the identity of that organism is kept in the DNA or the RNA. It's believed that we evolved from RNA. RNA was the first nucleic acid in the formation of life in the primeval soup. And then from RNA, we realized you can get much more stable genetic information in DNA. And that was the beginning of more evolution. But we go back to make RNA and then protein. So in plants, they found that there was a natural antivirus agent. They identified what it was, and it was a small sequence of RNA that formed like a hairpin, called a hairpin RNA. You know how a hairpin has two things that hold together? And initially it's straight line. Imagine taking the hairpin and opening it up. But because of the structure of those sequences, they stick to each other. So those small interfering RNA were found in plants, and what they did was recognize the RNA viruses, and they would then induce a set of enzymes that would chop up the RNA virus, and it wouldn't survive in the cell. So they began to explore for the presence of other small interfering RNA, and they found that all cells, all vertebrate cells, all mammalian cells contain in their cytoplasm siRNA. And it wasn't necessarily serving as an antiviral defense system. It was actually regulating messenger RNA expression. It was another feedback system that would allow the cell to do different things. So they began to realize that you could make a sequence of RNA in a test tube, put it into cells, and you could shut off certain genes. This led to a Nobel Prize by Mello and Fire and Mello. I like that name. It's two different people. One is Professor Fire, Professor Mello. Mello Fire. That led to a Nobel Prize in 1996. And it was very exciting because in this laboratory, you could turn off certain genes just for experiment. See, what happens if I shut off the gene for tyrosine hydroxylase? Well, you won't make any dopamine. So you could get a system where you can add siRNA designed to recognize the gene for a rate-limiting enzyme, so you get effects that you wanted. And then they said, this could be a great therapeutic agent for genetic disorders. So in the year 2000 was the first experiment. They showed that if you design small interfering RNA to recognize the messenger RNA for Huntington's, that hereditary degenerative disease, you could stop it. And in a mouse. And that's where I jumped in, it's been 3 years now, to design a way to deliver siRNA through a nasal route. And so this is what's going to happen with, if you can silence the alpha-synuclein that's prevented from spreading, and this is, I have some problems with the paper, but it's an exciting step forward. And I think let me just put it in 3 sentences. So it's very exciting now that we should be able to have a novel kind of drug therapy that will prevent the spread of Lewy bodies through the brainstem and the cerebral cortex, and thereby prevent the progression of the disease. It's something we've all wanted. I don't know when we'll have this in our hands to do studies in humans, but I think it'll be, once you find a benign way to deliver it. The reason this is this paper, they use a viral protein on their nanoparticle to make sure it attaches to brain cells. And then they put it in an exosome so you get across the blood-brain barrier. And once you start playing with viral proteins, giving it to humans, the FDA is not going to like it. Our approach doesn't use anything that the FDA doesn't like. So our approach, the intranasal route, you don't even need a needle, It just, and people say, well, isn't it unlikely that people that have lost a sense of smell would be able to take this medicine through their nose? I say, I'm not asking them to smell it. The nerves are still there. It's up in the brain that they can't interpret what those, isn't like you're losing those nerves. But you lose some interneurons that allow you to decipher what the smell means. But you haven't actually lost those nerve fibers that line your nasal cavity. So it should be able to work even if you don't smell anything. Okay, now I'll take questions. We're almost done. I thought that was I just saw this morning, and I've been thinking about it driving down. I said, how can I translate this highly technical paper to people? The message is, we're making really good progress in halting this disease in its tracks. And it isn't just a matter of giving levodopa, which, you know, we're still in the levodopa train. We still play around with all these variations of dopamine replacement. But as you all know, that's not the solution. It's good for managing your condition. It certainly improves your quality of life. It restores your lifespan to normal, because before levodopa, people would die much younger and in terrible discomfort. Now, at least, people can live to their expected life expectancy, to their normal life expectancy, as if they didn't have Parkinson's. The only thing that suffers is loss of independence, mobility, and those kinds of things. Susceptibility to falls, susceptibility to pneumonia, and that kind of thing. So I'll take questions.
There was a Michael J. Fox podcast that talked about this alpha-synuclein, and it mentioned that there was some human studies being done in Austria, and that this is being fast-tracked by the FDA.
You know, when I read the article, there was no talk about that. I don't believe it would be really fast-tracked because of the viral protein that it was required. You know, it may not be infectious, but they always worry about a reaction to that in humans. So, yeah, well, keep an eye. I'll have to look at the podcast, listen to the podcast. It would be exciting. I mean, I think it's worthy of trying to get this on quickly. This is a very big development. We've known about these they're called gene silencing molecules, siRNA. Now there's even a more stable form called antisense oligonucleotide, known as ASO. They're even better because they're more stable than the siRNA. And so you don't need to give the treatment as often. But whether it gets into humans quickly, that would be great. FDA never, the only things they will fast track is if people are dying left and right, you know, and it's a life-saving thing. So they're very cautious about things that may have unforeseen biological consequences. So they're there to protect people. So they tend to be cautious about certain kinds of treatments. For example, gene therapies have been around a long time, and then when they were using lentiviral vectors. So what I mean is you use a virus that you supposedly detoxify, that you make non-replicative, so it won't spread in your brain, and you take a bit of DNA out of that virus and you put in the DNA of the gene of interest, and then you infuse this into the person. First developed to treat these fatal leukemias in children. Well, people they didn't die of leukemia, they developed fatal encephalitis, a tremendous response to that viral coat. So in this report, they're using a lipoprotein, a coat from a virus, because these viruses have a neurotropism. They like membranes of neurons. So the two challenges, one is to get it from blood to brain. They put that in a special nanoparticle that permeates from blood to brain. But then once it's in the brain, it's got to stick to the neuron, and that's where they use the viral vector. Our method uses a manganese divalent chelate that allows it to get into neurons. And it uses a contrast agent that contains manganese that is approved by the FDA. That's why the manganese in high amounts can be toxic. The way that we prepare it doesn't. So I think what I want to do is get their siRNA against alpha-synuclein and package it in our system, and that could go faster. Anyway, there are ways that we can really fast track it. We'll get it on the superhighway. Yeah.
We met our doctor yesterday, and you spoke of pneumonia and Parkinson's, and we asked him when we got our last pneumonia shot, and he said in 2002. And I said, well, should we go ahead and get some now? And he said, well, I have read two different things on it. He said it used to be, they said after 5 years, you know, it was okay, or after 5 years, get another shot. Now they were saying, don't get a second shot, it doesn't do any good. And now they're reversing that. Have you read anything about that?
Well, what I've been told by the infectious disease people is that this flu virus mutates so often that you should get a new shot every year.
Is that for pneumonia? That pneumonia
No,
shot?
I'm thinking of the flu shot. For
Yeah, this is
pneumonia, I don't
pneumonia
know.
shot.
No, I'm sorry. I'm responding to the wrong vaccination. Yeah, for the flu, it's every year. For the pneumonia, I don't know, because it's a very specific kind of pneumonia. I don't know how frequent that should be.
So you haven't read anything in any
No.
of your papers about that?
No. But I'll look into it. See, I don't know every answer.
You have to give us a
That's
homework.
infectious diseases, you know. Whenever people have an infection, I send you back to your primary care doctor. To prevent against these pneumonias, the best thing to do is to make sure you don't have a lot of secretions, you get chest PT, you know, you kind of, and if there's a sudden worsening of your Parkinsonism over a day or two, usually it's an infection, most often bladder, secondly pneumonia. And for that, get your primary care doctor to see you quickly, do a urine analysis, listen to your logs, maybe even a chest x-ray. That's the extent of my work with infectious diseases. I see those as precipitating factors for an exacerbation of signs and symptoms of Parkinson's. The disease goes very slowly. There are people that have fluctuation day in and day out, motor fluctuation, but suddenly inability to walk or confusion, disorientation from one day to the next, worse dyskinesia, worse tremor, whatever, suddenly, without any change in medicine, usually is a red flag that you have some kind of physiological stress, which translates to some kind of infection. Right.
Can you, do you consider levodopa, carbidopa a mask where you, suppose you get better, you said that some people have gotten better or have eliminated
symptoms.
completely, yeah. Wouldn't that medication mask the progress?
Yeah, it does. And that's why whenever one wants to see if the disease is progressing or not, in clinical studies, let's say we're testing a new drug, drug X, and we give it, but we're not going to take away your levodopa because you'll be in really bad shape. You won't be able to function. So we keep you on that and adjust it as needed to keep your symptoms stable. And after 5 years, we like to examine you when you stop taking levodopa and compare where you were before you started the quote neuroprotective drug or not. Typically, you'll be in a worse shape because the illness progresses. The underlying process is not impacted by dopamine replacement. This is why we call it symptomatic treatment. We alleviate symptoms. But in a sense, it's masking the symptoms. Now, people like Muhammad Ali didn't want to take anything. He said, "I'll let it be. This is what the Lord gave me." And after a while, he needed to take it because he takes minimum amount of medication. Some people would rather let it run its course. If you're fortunate enough to have a slowly progressive form of the illness, those are what we call the tremor predominant form of Parkinson's, have a slower progression than the rigid akinetic form. But yeah, you're absolutely right. And a lot of people, I'm glad you brought that question up, because a lot of people think that the levodopa somehow is treating the underlying disease. What it's treating is one of the symptoms of the illness that is due to decreased production of dopamine, and we're replacing dopamine. And the reason that it gets worse and you start getting motor fluctuations is because the disease is progressing. In the beginning, you get by with 3 tablets a day, but the more dopamine terminals you lose, the less you can store of dopamine, so you need to give more levodopa more frequently. And then after a while, you can't give much more levodopa because you get overreaction, so you want to use a dopamine agonist that stimulates dopamine receptors tonically throughout the day, and that way you do better. And you don't have to rely on the remaining dopamine neurons to store the dopamine that is prepared from levodopa. So that's important to realize that the nigrostriatal system has 2 critical components. One is the dopamine neuron that projects fibers to the striatum, which is higher up in the deep gray structure of the brain, and then the striatum itself that has the dopamine receptors. The dopamine is the key, the receptor is the lock. Dopamine in the lock opens the door, you can walk. So if you lose dopamine, I can give you skeleton keys, synthetic keys, that turn the lock and allow you to walk. But if the lock is jammed, or there's something, piano behind the door, like that, then you won't know, all the dopamine in the world is not going to help. Those are the atypical Parkinsonism, or vascular Parkinsonism, where even replacing dopamine doesn't do a thing for those symptoms. And that's
a fraction of the total number of patients we see. Those are called the atypical Parkinson patients, where the problem is not so much in the lack of dopamine, but in the postsynaptic, or the target area, where the receptors are not responding appropriately to dopamine. Well, one more question I'm going to take, because I have to prepare for the clinic. Yeah?
I want to discuss with you one thing that you were talking about. When my wife first went on Asylink, she went on Synuclein because it was from Europe, and it hadn't been approved in this country. And the tradition has been that Europe approved medicines long before the FDA does. Have you considered using the European pipeline to get your stuff?
Well, that is. In fact, these studies are from Europe. The studies came from London and one other country, but the main authors are English. Yeah, it's true. They're more likely to approve drugs. They're quicker to approve drugs than we are. And the reason that the FDA continues to be really slow, they feel we escaped the thalidomide problem. Remember, thalidomide was a minor tranquilizer, an anti-nausea drug that was given to women who were having morning sickness. It was very big in Germany and in Europe. The FDA administrator didn't like some of the animal studies and said, we can't approve this drug in the United States. So thalidomide never made it to the United States. And the consequence of pregnant women taking thalidomide was phocomelia, which are kids who are born without limbs, just little hands coming out of the shoulders, and it was very sad. And there was a lot of people that were born because of thalidomide. So the FDA always says, see what because of our strictness, we've saved a lot of grief. So I mean, I can understand that, but that was in the 60s. The interesting thing about thalidomide is it had a rebirth, like so many drugs. Now it's being used for leprosy. And it's a great drug for leprosy, but you don't give it to pregnant lepers. They're already having their limbs falling off anyway. All right. I think that's it.